There are many subcellular components that you learn about in biochemistry that rarely get any more attention than simply knowing that they exist. Some of the structures that I've learned about year after year, with little to no added detail, are some of the structural motifs of DNA binding proteins. Specifically the classic leucine zipper, zinc finger, and helix-turn-helix motifs. Now normally I wouldn't be inclined to dive any deeper. That is, until I discovered an interesting article on PubMed that describes how epigenetic silencing of a tumor suppressor gene that codes for a leucine zipper protein could be a biomarker for ovarian cancer. So for the first time, I became interested in the lowly leucine zipper and decided to find out more. But before understanding the findings of the research, it's important to gain a preliminary understanding of the structure and function of the leucine zipper protein. As described in research articles by W.H. Landschultz and Dr. Francis Crick himself, the leucine zipper is composed of two right-hand alpha helices in a coiled-coil formation in the principal subunit of the protein. In this subunit, there is a leucine residue at every 7th position, and 3.6 residues per turn. This causes internal hydrophobic interactions between the leucine side chains, which stabilize the coiled coil of the alpha helices. These interactions connect the two helices in a sort of zipper-like structure. The N-terminal subunit of this protein is characterized by many basic amino acids, like lysine and arginine, giving the protein its more common name, the basic region leucine zipper. Such amino acids have a positive charge at physiological pH, and the two N-terminuses straddle the DNA major groove. Now, since DNA is negatively charged, due to the phosphates on its backbone, a strong electrostatic interaction occurs between the basic region of the leucine zipper and the DNA helix. Through these interactions, the leucine zipper is able to bind to DNA and act as a regulatory protein, activating or suppressing certain genomic functions. In this case, the leucine zipper plays a part in cell growth. With that in mind, let's go back to how all of this relates to the potential early detection of cancer. In the aforementioned article, the team of scientists analyzed ovarian cancer cell lines for the expression of the tumor suppressor candidate gene, LDOC1, which stands for leucine zipper downregulated in cancer 1. They used 7 cancer cell lines for this procedure and analyzed them via reverse transcriptase polymerase chain reaction, looking for expression of the LDOC1 gene. They used methylation-specific primers to look for methylation of the promoter regions. As they reviewed the results, they discovered that 3 of the 7 cancer cell lines showed a complete loss of LDOC1 gene expression. And as they predicted, the gene silencing was caused neither by gene deletions nor rearrangements, but by methylation and subsequent inactivation of the promoter region of the target gene sequence. This epigenetic methylation means that the cancer is not necessarily a genetic disorder, but could be due to heritable changes in the DNA that were not caused by changes in the DNA sequence. This could be due to mutations in methyltransferases, genomic mistakes in utero, or even due to general nutrition of the individual. But what was even more interesting was that they found if they added ADC, or 5-aza-2'-deoxycytidine, which is a known DNA methyltransferase inhibitor, the LDOC1 gene became reactivated. But what does all this mean, and how does it relate to the actual leucine zipper motif itself? Well, as mentioned before, the LDOC1 gene codes for a protein, the LDOC1 protein, which contains a leucine zipper domain. Now, this domain binds to other regions of DNA, which are responsible for cell proliferation, and that keeps tumor development at bay. If, however, the promoter region of this gene is methylated, it interferes with its ability to be transcribed to produce the helpful leucine zipper protein. This could let the cell grow uncontrollably, which would lead to cancer. The next logical question is, how does simple methylation of a few bases really lead to that much interference? Well, for that answer, we need to look at some information contained in a study published by Newell Price, Clark, and King. Their study shows that it isn't simply the added methyl groups that make it difficult for the LDOC1 gene to be transcribed, but it has to do with all of the changes that come next. Following methylation, binding proteins that preferentially recognize methylated DNA are often recruited. These proteins associate with histone deacetylases and chromatin remodeling proteins to increase the stability of the condensed form of the chromatin. In order for transcription factors to access their target genes, the chromatin must be in an open conformation. That is to say, spaced out enough so that they can fit into their target sequences, which in this case is the major groove of the LDOC1 gene. If, however, the chromatin is curled up tight and condensed, we say that it's in a closed conformation. This prevents the factors from interacting with the genome, and in the case of the LDOC1 gene, prevents the production of the LDOC1 protein to help control cell growth, which is essential for the prevention of tumors. Now, the mechanism relating the leucine zipper protein to cancer is actually quite simple. As we've learned, when the LDOC1 gene is transcribed, the LDOC1 protein can be made. And this protein binds to specific regions of the DNA responsible for cell proliferation. But there's another factor at play here, called nuclear factor kappa B. Active NF-kappa B turns on the specific genes responsible for the proliferation, and it appears that the leucine zipper of the LDOC1 protein binds to those DNA regions instead, preventing NF-kappa B from letting the cell grow out of control. So it makes sense that a methylated, untranscribable LDOC1 gene could have horrible consequences. The reason for all of the research done by the team investigating the epigenetic silencing of the LDOC1 gene is because ovarian cancer is categorized by a set of unspecific symptoms. It unfortunately is often diagnosed only at a late stage of the disease. Consequently, mortality rates are high because of this. So it's essential that we find better ways of detecting the illness before it has already taken hold. The results of this experiment are therefore not only scientifically relevant, but could have a huge impact on healthcare. Detecting methylation of these LDOC1 genes that prevent the leucine zippers from doing their job not only helps us better understand how our bodies work at the genomic level, but could also be a key factor in saving lives. After all, early detection and prevention are the best ways to fight disease.